Coming to active transport. Active in itself explains two of its characteristics. It is an energy dependent process and it involves movement of particles or ions or molecules from an area of their lower concentration to an area of their higher concentration. So this is against the electrochemical gradient or uphill. As moving up the hill, you need more energy, right? This is uphill or against the electrochemical gradient using energy. Now active transport is of two types: primary active transport and secondary active transport. Let's first talk about primary active transport. Primary active transport uses ATP as the source of energy, adenosine triphosphate. ATP is the source of energy in primary active transport, whereas secondary active transport uses sodium gradient. So primary active transport requires ATP, and the proteins that function in active transport are known as pumps. Now if you remember, proteins had a function in transport, and in transport proteins could act as channels, carriers or pumps. Channels we have seen in simple diffusion, carriers we have seen in facilitated diffusion. Now pumps play a role in active diffusion. Active movement rather. Okay, active movement, be it primary, be it secondary, the proteins involved are known as pumps. So in primary active transport, the most commonly involved protein or most commonly involved pump is the sodium potassium pump. Now the sodium potassium pump is present in the cell membranes of all the cells of the body. It is present in all the cells of the body and it plays a vital role in maintaining the homeostasis of the body. Now this is the structure of the sodium potassium pump. Okay, before we understand how it functions and what is its significance, let's see a little bit about the structure. So the sodium potassium pump consists of two subunits: the alpha subunit, which is a bigger one, and the beta subunit, which is a smaller one. The molecular weight of alpha subunit is 1 lakh and the molecular weight of beta subunit is 55,000. Now the actual function of the beta subunit is unknown, but it is known that the beta subunit is attached to glycoprotein residues. Okay, coming to alpha subunit. The alpha subunit has various sites for attachments of various molecules. This is the inside of the cell and this is the outside of the cell. This is the cell membrane. So on the outside, the alpha subunit has sites for binding of 2 potassium ions. How many? 2 potassium ions, and a site for binding of ubiane. Now what is ubiane? It is a drug that is used in heart failure and it causes inhibition of the sodium potassium pump. That's all you need to know. It is not really significant, but it is mentioned here because ubiane was used in heart failure and it caused the failure of this pump, which is vital for maintaining homeostasis. So this is now no longer used, right? This has a site on the outside of the alpha subunit. So outside has 2 sites for binding of 2 potassium ions and ubiane binding site. On the inner aspect, it has a site for binding 3 sodium ions, and it has an ATP binding site with ATPase activity. ATPase activity cleaves ATP into ADP and inorganic phosphate, releasing energy. Now you can see where ATP is used and why this is called as a pump, as the primary active transport. Active because energy is used. The energy is in the form of ATP, therefore it is primary active transport. So this is the sodium potassium pump, which is an example of primary active transport. Now let's come to the functioning of sodium potassium pump. Now how this functions is that intracellularly 3 sodium ions bind to the sodium binding site, ATP binds to the ATP binding site, and outside extracellularly 2 potassium ions bind to the potassium binding site. When all these 3 molecules bind to their respective sites, a conformational change takes place, and with the ATP's activity, ATP is cleaved. Remember again, sodium binds, potassium binds, ATP binds. Now ATP is cleaved by ATPase activity. The energy released causes conformational change, which pumps these 3 sodium ions outside and 2 potassium ions inside. So what this pump does is pumps the 3 sodium ions to the outside and 2 potassium ions to the inside of the cell using ATP as source of energy. So this is the functioning of the sodium potassium pump. Now what is the significance? What do you achieve by pumping 3 sodium outside and 2 potassium inside the cell? What happens? So this leads us to 3 functional significance of this pump. The first significance is that it maintains the sodium potassium concentration inside and outside the cell. Now normally in a normal cell at rest, this is the cell membrane and this is the sodium potassium pump. Inside the cell, the concentration of potassium is much higher compared to the outside, normally under resting conditions. The concentration of sodium is higher in the extracellular fluid, that is extracellularly compared to the intracellular fluid. This is normal scenario. Now as we all know that the cell membrane is semi-permeable to both sodium and potassium, but more to potassium. So what happens is that potassium keeps leaking out because it is more on the inside and less on the outside. So it tends to move along its concentration gradient. So potassium tends to move outside and sodium tends to move inside because sodium is more on the outer aspect compared to the inner aspect. So if potassium keeps on moving outside and sodium keeps on moving inside, the normal homeostasis that the cell has maintained will be lost, right? Because we want sodium to remain outside and potassium to remain inside. So what role does sodium potassium pump play here? What happens is that when sodium comes inside and the insides of the cell reaches a particular level of sodium, it activates the sodium potassium pump, which will pump the sodium outside. Similarly, when the concentration of potassium reduces below a critical level, this activates the sodium potassium pump, which will pump 2 sodium inside, thereby maintaining the concentration of potassium inside. So this is the maintenance of sodium potassium concentration within the cell and outside the cell. Now just for your knowledge, the levels of sodium and potassium, they might be asked as MCQ. So sodium in ECF and ICF and potassium in ECF and ICF. As you all know, sodium is mainly present in the ECF, so its concentration is 142 milliequivalents per liter. Inside the cell, sodium is present as 14 milliequivalents per liter. Potassium is the main cation intracellularly. Its concentration is 140 milliequivalents per liter, and outside the cell its concentration is 4. Now the most important point to note is that the ratio of sodium ECF to ICF, or rather ICF to ECF, is 0.1. This value you should remember. And the ratio of potassium ICF to ECF is 35. This is the normal ratio. So to maintain these normal levels, the sodium potassium pump is important. The second point we need to know, the second functional significance of this pump, is maintaining the cell volume. Now what happens is that this is a cell. It has various ions, like we said, sodium, potassium, etc. But as we all know, within the cell there are many proteins. These proteins cannot pass through the cell membrane, so they remain within the cell. Moreover, these proteins are negatively charged. They are negatively charged. So what they do is they attract more cations around themselves. They attract more cations around themselves, and because of this, the number of ions within the cell will increase, obviously. So if the number of ions within the cells increases, this causes increased osmotic pressure, so water will move in, right? So if water moves in, this cell will start to swell. Its volume will be disturbed and eventually the cell will burst. So sodium potassium pump plays a very important role in maintaining the cell volume and prevents it from bursting. How? Now as we all know, sodium potassium pump pumps 3 sodium outside and 2 potassium inside. So as you can see, there is a net loss of 1 cation or 1 molecule on the inside. 3 sodiums are pumped outside and 2 potassiums are taken in. So a net loss of 1 molecule or 1 cation is there within the cell. So what this does is this reduces the total number of molecules within the cell. So the total number of molecules within the cell reduces, the osmotic pressure within the cell will reduce, and the cell volume will be maintained. The water won't move in. So this is the second function of sodium potassium pump. The third function is that it acts as an electrogenic pump. Now what do you mean by that? Again the same concept. The sodium potassium pump pumps 3 sodium outside and 2 potassium inside. As we saw here, there is a net loss of 1 molecule. Similarly, there is a net loss of 1 positive charge on the inside. Because if 3 positive charges are moving out and 2 positive charges are moving in, a net loss of 1 positive charge is seen on the inside. So this creates slight electronegativity on the inside and electropositivity on the outside. So this is the electrogenic pump action. Now this entire significance or the presence of negative charge within the cell at rest will be explained under resting membrane potential. As we all know that at rest the insides of the cells are negative and a role is played by the electrogenic pump activity of the sodium potassium pump. So this is the activity, another functional significance of the sodium potassium pump. Now another word that is important to describe the structure is heterodimer. Hetero means different and dimer means two. So this basically refers to the structure of sodium potassium pump. As we can see, it is made up of 2 parts, 2 subunits, and they both are different. One is alpha, one is beta. So this is the structure, the mechanism of action and the functional significance of the sodium potassium pump. So that is all about the primary active transport. Secondary active transport utilizes the sodium gradient as the source of energy. The sodium gradient, as explained earlier, is the difference in the concentration. Now we saw that inside the cell potassium is more and outside the cell sodium is more. So naturally sodium tends to move inside the cell. So this is the sodium gradient. There is difference in the concentration of sodium within and outside the cell. Now this is a cell and sodium is at a higher concentration on the outside, right? So sodium tends to move in. Now as sodium moves in through a protein here, as sodium moves in, other substances tend to join it. They move in along with the sodium or they move to the opposite direction, that is outside. Now remember, sodium tends to move inwards because it is in the higher concentration on the outside, so it will move inside. So if a substance, any other substance, say glucose or amino acid basically, if it moves along with sodium within the cell from outside to inside the cell along with sodium, this is called co-transport. Co means together they move. Together they move within the cell. This is co-transport. But you may ask that secondary active transport. Active means movement from low to high. Sodium is moving from high to low. So how is this active? Now this is active not in relation to sodium. This is active in relation to this substance which moves along with sodium. Now remember, sodium movement, sodium gradient is just the source of energy. The active transport or the substance that is transported actively is this substance, which may be glucose or amino acids in co-transport. They move together. So when glucose or amino acid move along with sodium within the cell, they do so against their concentration gradient, which means that glucose and amino acid are present in more concentration or higher concentration within the cell compared to the outside. So if sodium moves along its concentration gradient, it is the sodium gradient as a source of energy. But glucose or amino acid, if they move along with sodium in the same direction, that is co-transport, the direction is same but the concentration gradient is the opposite of sodium. Do you get me? Sodium moves along the concentration gradient but glucose or amino acid move against the concentration gradient. Therefore it is an active transport, and since it utilizes sodium gradient, it is a secondary active transport. And since they move in the same direction, it is co-transport. Coming to counter transport, counter means opposite. So there are certain pumps in the body. Now remember, these channels with these proteins which operate in active transport are known as pumps. There are certain pumps known as sodium hydrogen pump or sodium calcium pumps. Now for example, the sodium hydrogen pump, when it functions, sodium naturally tends to move along its gradient, But since this is counter transport, the molecule have to move in opposite directions. So hydrogen definitely has to move out, right? And hydrogen does so against its concentration gradient. Since the movement is opposite, it is counter transport. Since hydrogen is moving against its concentration gradient, it is active. And since it utilizes the sodium gradient, it is secondary. So this justifies each word I have explained, that how this is moving. So these two pumps, the sodium hydrogen and sodium calcium, are examples of counter transport, whereas glucose and amino acids, they move along with sodium, and these are examples of co-transport. So that is all about the active transport, the primary and the secondary, co and counter transport.